Hey guys, welcome to video 28 for Mr. McGee's 9th grade biology course at Arizona School for the Arts. Today's topic is RNA and protein synthesis. We'll be learning about how the information encoded in a cell's DNA is used to create proteins. First, let's remember that DNA is a double helix composed of long strands of nucleotides. Between the two strands, base pairing occurs, where H bonds between the complementary between the complementary nucleotides form rungs of the DNA ladder. During replication, those base pairs are split by helicase. This allows the DNA polymerase to add new nucleotides to each strand using the original strand as a template. And because A only pairs with T and C only pairs with G, proofreading that strand is easy. Mismatched nucleotides are just rejected, and the correct base pair is made. So we're going to see that protein synthesis also uses base pairing to ensure that the information in a gene is correctly copied and translated into a functional protein. RNA is a nucleic acid, very similar to DNA. RNA is a long chain of nucleotides like DNA is. RNA carries genetic information. However, there are 4 important differences to remember. First, RNA is single-stranded. Secondly, RNA can leave the nucleus. DNA can't do that. RNA is composed of the phosphate and pentose sugar and a nitrogenous base, just like DNA. But in RNA, the sugar is ribose instead of deoxyribose. And the nitrogenous bases in RNA are adenine, guanine, and cytosine, similarly to DNA. But RNA has the nitrogenous base uracil instead of thymine. RNA comes in 3 types. There's messenger RNA, which is a temporary copy of a working gene that carries the genetic information from the nucleus to the ribosome. There's transfer RNA, which takes amino acids to the ribosome so they can be made into polypeptides. And then there's ribosomal RNA, which is part of the structure of a ribosome. Protein synthesis is a process that involves all 3 of those RNA types and also DNA as a permanent copy of a gene. So we'll break down protein synthesis into 2 parts. The first part is transcription, in which the gene is copied as a molecule of mRNA that then leaves the nucleus and goes to a ribosome. And the second part is translation, where the ribosome uses the mRNA as instructions to join amino acids together, carried by tRNA to make a polypeptide. So let's start with transcription. In some ways, transcription is similar to DNA replication. It takes place inside the nucleus. It involves splitting the base pairs in the double helix. But the goal of transcription is very different. While the goal of replication is to create 2 identical chromosomes in their entirety, the goal of transcription is to create a temporary copy of a working gene that can leave the nucleus and then be used by a ribosome. So transcription begins when the enzyme RNA polymerase unzips the double helix. It does that by breaking the H bonds between base pairs, just like helicase would in DNA replication. This leaves the sequence of nucleotides on each strand of DNA exposed, and only one of the 2 strands is then copied by RNA polymerase as a little piece of mRNA. That copied portion of the DNA is called a coding region. A coding region is often what we refer to as a gene. After it has separated base pairs, RNA polymerase can read the coding region and add ribonucleotides in the proper order to make a complementary strand of mRNA. RNA polymerase works similarly to DNA polymerase by adding nucleotides to create base pairs according to Chargaff's rules. But of course, RNA polymerase adds ribonucleotides and not deoxyribonucleotides, and it only forms base pairs between C and G and then adenine and uracil instead of using thymine. Another difference is that RNA polymerase only copies a small part of one of the chromosomes instead of the whole thing. When it's finished, the new piece of mRNA will detach, it leaves the nucleus, and then after transcription is through, the DNA strands just reform their H bonds that were separated by RNA polymerase. You might be wondering how RNA polymerase knows how to find the right gene to copy, or for that matter, how it knows when to stop. Well, the answer is in the DNA itself. Somewhere before the start of every gene, there's a region of DNA called a promoter. The promoter tells the enzymes involved in transcription where to start. A common promoter is called a TATA box because it consists of some variation of the nucleotide sequence TATAAAA. At the end of every gene is a similar code that tells the RNA polymerase to stop copying the DNA. Hey, that's a lot of science. It's time for a joke. So, what did the biologists name their twin baby boys? Ready? John and control group. Okay, after transcription occurs, but before the mRNA has left the nucleus, most mRNA molecules need to be edited because they contain regions that are copied from the DNA template that are not actually used for protein making. These small little non-coding regions are called introns because they're found inside a gene. They're in the way of the gene. Enzymes in the nucleus cut the introns out of the mRNA, leaving behind only the regions that are involved in making a protein. Parts of the gene that are used to make proteins are called exons because they're expressed. Once the introns are removed, special enzymes splice the exons together to produce a mature mRNA molecule that can then leave the nucleus. Remember that genes are the instructions for making proteins, and when a gene is copied as mRNA, those instructions are used by a ribosome during translation to specify the precise sequence of amino acids needed to produce the primary structure of a polypeptide. Right? That's the sequence of amino acids in a polypeptide. So let's examine how the sequence of nucleotides in mRNA is used to specify the sequence of amino acids that is needed. The genetic code is composed of 4 different letters, A, U, C, and G. A quote-unquote word in the genetic language is composed of 3 of these letters in sequence, like AUC or AAA. So the sequence in the mRNA is called a codon. A codon is what specifies a specific amino acid. For example, the codon that reads CAC in mRNA specifies the amino acid histidine, while a codon that reads UCC specifies the amino acid serine. Behold, the codon decoder wheel. Alright, so it's not that exciting, but this wheel is really important because it allows you to interpret the gene encoded in mRNA and determine the sequence of amino acids that a piece of mRNA codes for. The innermost circle of the wheel represents the first letter in the codon. The next circle is the second letter of the codon, and the third circle is the last letter of the codon. The outer circle is where all the amino acids are, and that tells you which amino acid is specified by that sequence of 3 letters. So let's practice using the codon wheel to find out the right amino acids for these codons. What amino acids are specified by the codons AAC, GAU, CCA, and UUU? I'm waiting. Hector, begin work now. Alright, so you should have gotten the amino acids asparagine, aspartic acid, proline, and phenylalanine. Is that what you got, Hector? Don't lie. That's not what he got. He got something totally different. I don't think he understands it. Hector, you need to come in for tutoring. Alright, but now let's look closely at the codon wheel, and you notice that many of the amino acids are specified by more than one codon. So there's lots of different ways, for example, that we can make arginine. Arginine has no less than 6 different codons. By the way, arginine is the pirate's favorite amino acid. Because it's arginine. Because it's arginine. Arginine. Get it? Okay, also, you might have noticed that there are codons such as UGA, UAA, UAG that code for the word stop. Stop is not an amino acid that I know of, but stop, when coded by these codons, tells the ribosome when to stop adding amino acids and when it's time to release the new polypeptide. That's done. Stop means stop. It's done. There is also a codon that tells the ribosome when to begin. That codon is called the start codon, and it's the codon AUG, which codes for the amino acid methionine. So every single polypeptide begins with the amino acid methionine. Neat, huh? All right, finally we get to transcription. translation, which is where tRNA is going to bring amino acids to the ribosome so they can be added to the polypeptide. And this process itself is pretty complicated, so we'll probably cover more of it in class. But I do want you to know a couple of things. First of all, each tRNA molecule has a region called an anticodon. That anticodon is composed of nucleotides that are complementary to the codon for the amino acid that is specified by the mRNA. So for example, an mRNA may have the codon AUG, and then the anticodon would be UAC, and they would match up. And then the tRNA that has the anticodon UAC would have stuck to it methionine, which is what the codon AUG specifies for. Right? Does that make sense? Probably not. We'll work on it. It'll come to you. You'll be inspired one of these days. Anyway, tRNA then briefly forms base pairs with the codon in mRNA, while the ribosome does its work, which is to transfer the amino acid that tRNA brings to the ribosome to the growing polypeptide chain. If the codon for threonine is ACA, then only tRNA with the anticodon UGU will pair with it, right? Just like base pairing. It's base pairing. Base pairing codons to anticodons allows the ribosome the ability to add amino acids in the right sequence according to the instructions in DNA. But again, translation is fairly complicated. We'll go over it more in class. For now, though, that's the end of this video. Please complete the corresponding review questions, be prepared to review this material in class, and wait for it! Bunny of the day.